Hi everyone, my name is Annie. I'm a PhD research fellow at the University of Oslo in Oslo, Norway. I work at the Lichenology at the Natural History Museum with the Integrative Systematics of Plants and Fungi group. Today I'm going to introduce one of my projects that is still in progress using stop-motion animation. My research focuses on the integrative taxonomy of the lichen family Psoraceae. I'm using integrative taxonomy because it combines different information about species, such as the morphology, DNA, chemotype, and ecology, in order to better understand things like biodiversity, conservation, evolution, and applied sciences. In this project, I'm studying the Psoraceae because the existence of the Psoraceae is a bit uncertain. In 2011, Ekman and Blahlied published a phylogeny of the family, investigating the circumscribed genera at the time, including Psora, Protoblastinea, Micaria, which was later changed by Ekman and Svensson to Bryennaria, also Glyphopeltis, Psorula, Eremastrella, and they included some species from other families, such as the Pilocarpiae and Ramalinaceae. So the question still remains, what is Psora, what isn't Psora? So here is some background on the Psoraceae. The Psoraceae is established as the sister to Ramalinaceae, at least it has been shown by other people multiple times, in the Lecanorales, the Lichenomycetes in Ascomycota. The family was first described by Zaltbruckner in 1898. He later synonymized the family with Lichenology and then resurrected. The family was resurrected by Halffelder in 1984, and he based it on the apical apparatus of the ascus. The name Psora was first used to describe lichens in 1789 by Hoffman, and the name was brought back in 1980 based on usage from 1796. Psora decipiens is the type species, and there are more than 30 species that have squamulous thalli, calcium oxalate crystals in the hypothecium, and a Psora-type ascus. Some Psora species have secondary compounds in the thallus, and all have anthraquinones in their apothecia. Psora live in arid regions on calciferous soil and rock. They associate with the photobiont Eremastrella and potentially others. They also have been well studied with over 1,000 sequences available on NCBI. Protoblastinea, the sister genus to Psora, was first described in 1772 by Scopoli as lichen rupestis. It went through a series of different names until it was raised to a genus level in 1911 by Steiner. There are 30 species that have crustose to areolate thalli, emarginate apothecia, and Psora-type asci. Like Psora, there are anthraquinones in the apothecia. They have a worldwide distribution on calcareous rock, and they associate with a chlorocoid photobiont. There are 75 sequences publicly available on NCBI. The genus Bryennaria was named for the Micaria sylvicola group in honor of Brian Coppens. There are 4 species with areolate thalli, emarginate Ascomycota, and Psora-type asci. Bryennaria has not been found with secondary compounds in the thalli or with anthraquinones in the apothecia, but there are some other pigments that are found in the apothecia. Bryennaria grow in shaded acid rock areas associated with non-micarioid photobionts, and there are 10 sequences currently available on NCBI. The genus Psorula has an uncertain position in the family Psoraceae. It was first described by Tuckerman as Boyatora in 1848, and Schneider raised Psorula to genus level in 1980. The only species, Psorula rufonigra, has a squamulous thallus, Psora-type asci, and is distinct from Psora due to its dark brown hymenium. Psorula is not known to have secondary compounds in the thallus and does not have anthraquinones in the apothecia. It tends to grow on spirulina in arid regions and associates with a chloroplacoid algae that my initial results suggest might be Travoxia. There are currently 3 sequences available on NCBI. At this point in my research, I do not have enough information to circumscribe the family Psoraceae. There is evidence that Psora, Protoblastonia, and Bryonaria are well within the family, and there is also evidence that Psorula, Protomicarea, Glyphopeltis, and Eremastrella are outside of it. To circumscribe the family, I'm going to test the diagnostic morphology of the genera. I'm also going to sequence additional DNA from multiple specimens in each genus. I plan to test chemotypes for the genera, as well as investigate the habitat preference, in addition to the photobiont preference and potentially additional symbionts that may associate with the lichens.